and a very good day to Dr. Wong and everyone. We are from Group 2 Section 2. We'll be presenting about the blood pressure method, which is the volume clamp method. Next. Okay, so far we will be having a content which is introduction, importance, mechanism, strength and weakness, and also the feature view, which is the challenges and commercialization. Okay, for the introduction, this volume clamp method was introduced by Czech physiologist Jan Penas. What is this method? Which is the process which use a control loop to manage the pressure in the clear side finger cuff in order to control the volume of the arteries and directly measure the finger cuff pressure in order to display it as a waveform. This method also known as a vascular unloading techniques. This method is continuously and non-invasively method which measure the blood pressure using the finger arteries. This traditional method is to convert light to become a weak current to collect the signal of blood volume, the blood volume pulse by a sensor. And also the main component of this device is a photoplethysmography sensor as the light emitter. The second point is the importance. This method will provide a real-time continuous beat-to-beat blood pressure, cardiac output, stroke volume variation, and also the systemic vascular resistance. And also the importance is to measure the finger arteries diameter by using an implantable finger cuff containing an infrared transmission plethysmography. And then it also maintain the constant arterial size and transmural pressure in the finger so that the reading will get more accurate. However, the frequently movement of finger can disturb the reading of this method of blood pressure. Okay. Next, we will cover.
Okay, you guys can hear my voice?
Yes, Shukri.
Okay, for the photoplethysmography sensor is a main component of the volume clamp blood pressure. According to the Castaneda D 2018, in their experiment show that there are two types of light, which is infrared light and the green light. For the infrared light function is more to emit the light which is into the deep concentration to the body part to get the measurement of the oxygen, and then for the green light as the main source of the light, which is can calculate the observation oxygen either in oxyhemoglobin or deoxyhemoglobin. And in their research also, according to the Chan Cooper 2019, this type of photoplethysmography sensor can be combined with the electrocardiography, which is for the electrocardiography function is to determine the state of systolic, where the photoplethysmography sensor is used to determine the time taken for the pulse wave, which is the pulse wave is measure when it start from the heart to the patient finger. Okay, next.
For the mechanism of the volume clamp method, first the cuff is placed on the finger. After that, the blood volume is measured continuously by the light emitted diode or LED and the photoplethysmography PPG sensor. And after that, the cuff will automatically inflate or deflate according to the blood volume detected to keep the blood flow constant. From the change of the cuff pressure according to blood volume, from the change of the cuff pressure according to the blood volume, an algorithm is used to produce the blood pressure signal. Next, from the figure show, we can see that the cuff contain the infrared transmission and plethysmograph sensors, so that the cuff will measure blood the blood volume. And after that, the cuff will inflate or deflate continuously. So after that, the signal of the cuff pressure needed to keep arterial blood volume constant is in, and then after that go to the control system or algorithm, and lastly the continuous non-invasive blood pressure measurement is get. Next.
Hello, thank you, Rawani, for the explanation. So what you're seeing right now is the calibration process and also the working mechanism of the volume clap method. These three signals are reference to the finger arterial pressure, the first one. The second one is the PPG or photoplastograph signal from the diode. And this one is the pneumatic pressure from the pressure valve that control the pressure of the cuff. So there's sections here like A, B, C, and D. Let me just explain a bit one by one. So in A, the cuff pressure is smaller than the arterial pressure. That leads to small PPG posality, means that the signal of the PPG signal is small, which is, we don't want that. So in B, in part B, the cuff pressure reaches the arterial pressure, which means the cuff pressure, the dynamic value of the pressure valve, it's reaching the value of the arterial pressure. So leading to zero transmural pressure. Transmural pressure means the difference between the arterial pressure and the cuff pressure. So it's best to reach zero transmural pressure, thus maximizing the PPG posality, means the PPG signal, so like it peaks. So in C, the artery is unloaded. The term is unloaded. It means when the pressure inside the arterial pressure equals the cuff pressure. So that term we call the artery is unloaded. So in D, that's when the calibration phase ends. So in D, where we see the PPG signal, this refers to the constant blood volume. So like in the blood, it's constant all around. So the pressure valve will continuously clamp and like control the cuff pressure to be similar to the arterial pressure. Therefore, the pressure of the pressure valve will equal to the pressure of the arterial pressure. So you will see that the reading are similar. So that's the working mechanism of the volume clap method. Next, please. So in here we have a video. There's an product that utilizes this method called FinaPress, finger arterial pressure. So we would like to show you how it is in video form.
FinaPress develops medical devices and software for continuous, non-invasive blood pressure measurement and hemodynamic monitoring. The system functions via the volume clamp method, developed by Professor Peniers in the 1970s. An inflatable cuff exerts pressure on the finger, while the light sensor monitors the diameter of the finger arteries throughout the blood pressure pulse cycle. Initially, it will assess the artery to determine its unloaded state. Subsequently, the nanocore module will start measuring blood pressure using the physiological algorithm. The nanocore processes the feedback from the light sensor and dynamically controls the pressure inside the finger cuff to keep the diameter of the arteries unchanged at an unloaded state. Consequently, pressure inside the cuff that is needed to maintain this equilibrium resembles the arterial blood pressure in the finger. The finger press nova also calculates brachial pressure from the measured finger pressure. An arm cuff is used for only a one-time calibration of this calculated brachial arterial pressure.
Okay, so that's the video of one of the devices that utilize volume clamp method. So next please, Lisa. All right, so this is an so this is an example of what an arterial pressure graph looks like. So there is a starting point of the systolic uptake, and then there's the peak systolic pressure, where this is systolic reading, and then there's systolic decline, and then there will be a notch, and then the diastolic starts, and then at the end here is the end diastolic pressure. So like this whole the height of this graph, amplitude graph, is the pulse pressure, and the area under the curve is the mean arterial pressure. So yeah, this is roughly what would like an arterial pressure graph looks like. The reading will be taken the above and also the below, systolic and dystolic. So yes, my colleague will continue after this on the strength and weaknesses.
Okay, now I'm going to proceed with strength and weaknesses of this method, which is volume clamp method. Okay, for the strength, the first point is provide continuous estimate of the pressure pulse waveform. This is due to the instantaneously applied pressure, which is correspond to the artery unloaded pressure at each phase of cardiac cycle. So the second one is non-painfully. The third point is pressure generated at the cuff is equal with intra-arterial pressure in terms of efficiency. So which means during calibration phase, the cuff pressure will be adjusted to achieve maximum amplitude of PPG pulsatility along the cardiac cycle. So if the cuff is applied intra-arterial pressure, it will cause the artery become over-distant, become stiffer, and also decrease in pulsatility. In other side, if the cuff is placed below the arterial pressure, it will cause artery become collapsed and also decrease in pulsatility too. Therefore, with the help of this clamping, the volume of the artery can be at maximum PPG pulsatility, and the pressure become equal with the intra-arterial pressure, like I said before. So next, for the weaknesses, my friend will further explain. Yeah.
So for the first weaknesses, this method cannot be used in many situations. For example, like to those who has hypothermia and vascular disease, the blood flow in the figure can be very weak. It can cause unstable reading. The next one, for the accuracy, it does not meet the international standard for blood pressure measurement. BSH and AMI stand for British Hypertension Society and US Association for the Advancement of Medical Instrument. And the next weakness is the problems associated to the automatic calibration and overestimation of the systolic pressure. Thank you, that's all.
Okay, thank you for the presentations. Now we have Q&A section. Any questions? Yes.
Haven't finished it yet. Got one more content.
Okay. So, Fivio, come on.
Okay, my name is Ammar. I'm going to explain about the challenges. So, for this type of measurement and based on the studies that we have prepared our work on, there were few limitations that have been faced or affected the measurement, or we can call it challenges as well. So, first point, continuous non-invasive devices are all sensitive to patient movement. Example, in the case of severe vasoconstriction and peripheral vascular diseases or disordered fingers due to arteries, clinical experts have shown that it may be difficult to obtain a valid waveform using finger cuffs. Another point, problem with patients which have low peripheral blood flow. This is because the control system will become unstable when vascular diseases or hypothermia are being faced. The blood flow of the finger can be very weak. Third point, some patients report discomfort from this congestion in venous return from the fingertip where the cuff is placed. How to overcome these challenges? Monitoring of the conscious patient is possible, but measurement results need to be checked for plausibility. Upgrading the control system to become more stable. Also, manufacturers recommend changing the cuff to another finger after a certain period of monitoring. Now my friend Badro will explain the commercialization.
Okay, hello. The chance for commercialization is actually quite attractive for this volume clamp. This is due to the fact that mostly it is made of cheap materials like plastic or Velcro, and most of its electronics can be mass produced cheaply as well. It is also very easy to use because of its small size and very straightforward application. And for efficiency's sake, it has the same. It can detect the same intra-arterial pressure as in our fingers. The company Edwards Life Sciences actually has made a product, a very lucrative product called ClearSight System, and from there we can see that the chance for commercialization is actually quite good. It also has support for mobile apps for display and as well so that patients can monitor the actually healthcare staff can monitor the blood pressure through using mobile apps. That is all from our group. Thank you very much.
Okay, thank you. Now we open for Q&A section.
I have a question. I want to ask you guys regarding the weaknesses in which the accuracy. So can you guys show this? Okay, for the accuracy, you guys mentioned that it does not meet the international standard. And based on your mechanism explanation, you have explained truly in concise manner on how the mechanism is accurate to be able to obtain continuous blood pressure monitoring. So can you clarify in terms of accuracy, what does it mean by it does not meet international standard? Is it, for example, the result that is obtained, is it less than required standard, or is it it have a percentage error or something like that? So can you clarify this part here? Thank you.
Okay, thank you for the questions. For the weaknesses for the accuracy, it said that it does not meet the international standard due to the underestimate result, which is according to the BHS and also AAMI, supposed to be the reading is 5 plus minus 8 mmHg. So for this, they didn't mention the specific result that they did not meet the international standard, but they only state like that. But somehow the supposedly measurement is 5 until 8 mmHg. Yeah, that's all.
Okay, all right. Thank you for
Okay.
answering my question.
All right, welcome.
So any other question?
I think got one in chat, doctor.
Yeah?
There's a question in chat, doctor. Let me just answer that. Jahid just asked, does putting it in the right hand or left hand will cause any significance? From the research that we found regarding this method, they didn't specify, not say specify, but like they would usually go for the right hand, the index finger on the right hand to measure the arterial pressure. But regarding your question, this is an opinion based. I don't think that it will bring any significance, heavy significance to the reading, especially since in our slides of overcoming challenges, it is possible to put the sensor in other fingers as well to measure the arterial pressure. So that will be our answer for this question. Does that answer your question? Ong? Right. Thank you. Anyone else would like to ask?
Yes, anyone else? So if no, then that's it for today.